Well, good morning, everyone. I'd just like to reiterate what Terry has mentioned early on, and that is welcome to our webinar series. And what we've been able to do, what we have done is to declare this our science roundup. And this webinar series is a surrogate for the research forum that we normally host annually in April of each year. But due to the current challenges of associating in purpose, this webinar series is the best next thing. We're thankful that speakers were able to reschedule to speak during or for this webinar series and take time out of their way to be with us. Certainly their schedules are very dense, and we've had a number of people that just simply can't manage all of the necessities that are on their plate right at the moment. But we have a roster of people lined up, and what you will see is if you attend all our webinar series, and I invite you to visit the FRI website and register for each one. This is important because when you register, you automatically receive the link to join the webinar, and you also get a reminder. So that's very important. So, and I also want to just make one little correction here. And Terry had mentioned that we are the speakers. Yes, we are speaking, but Antonia is the main speaker. She's the main attraction. And I'm just providing you with a little bit of upfront information which will allow you to view the full roster of presentations that are going to be presented. And as many of you know, the Mountain Pine Beetle program is just one of many programs carried out under the auspices of FRI research. And I would like to say that this is why we do what we do. And these are a series of photographs that I use quite frequently because I just believe that they just emphasize the importance and the severity of the issue that we're trying to address. The upper left-hand photograph taken by Lorraine McLaughlin in 2006 was taken in the Chilcotin Plateau, and you can see that the entire forest area there is not green. It looks pretty red to me, which would suggest that this was a very severe outbreak in the Chilcotin. And over time, this outbreak migrated westward and south in British Columbia. And finally, in 2011, another picture was taken by Don Poblubny in the Grand Prairie area. And you could see that as a result of the overflight in 2006, that the pine-dominated forests were very severely impacted. And also the bottom right-hand photograph is also in the Grand Prairie area. And this was taken in 2014. I spoke to Brooks Horn, and he believes that he took that photograph, but we struck a balance, and we gave a photo credit to Sustainable Resource Development and Agriculture, Alberta Agriculture and Forestry. But you can see how widespread this is, and this is exactly why we do what we do. So the real start in Alberta, although some would argue that it happened even well before 2006, and it has in southern Alberta, but there was an overflight from BC into northwestern Alberta. And about that time, Sustainable Resource Development, the Forest Health section, under the leadership of Daniel Lux, where he was really had his entire section very much focused on developing an Alberta mountain pine beetle strategy. And this strategy sought to reduce risk posed by the mountain pine beetle. But there was also so much to be concerned about in addition to just the loss of forests. And what I mean by that is that with the loss of forests, we lose ecological services. There's impacts on wildlife habitat, water quality, and the like. And so there was much to be really focused on as a result of this strategy. And the current strategy is being shouldered by Erica Samus, who is now the director of the Forest Health section or the adaptation section, and her staff have been doing an amazing job in taking science that is coming along as a result of research and putting it into practice and influencing operational decisions and certainly policy development. Now, the primary objective was to contain the infestation and control the spread of the mountain pine beetle north and south along the eastern slopes of Alberta, and also to prevent the spread eastward. But I think that over time, we've come to realize that it's very difficult to contain, and it's certainly almost impossible to prevent. I've heard words like eradication, and certainly eradicating the mountain pine beetle isn't going to happen. I just don't see it moving in that direction, because once it's there, it's there. It may decline in its populations may decline significantly over time, but to eradicate it, I don't think it's going to happen. Not in our life, not my lifetime. And the FRI research initiated a mountain pine beetle program in 2007 under the auspices of the Foothills Model Forest, and it was to act as a science information forum. And its main purpose was to facilitate knowledge transfer, collaboration amongst researchers and managers, and also to identify information needs or research needs. And the mountain pine beetle program goals were guided by a terms of reference, a research prospectus, and a program activity team, which was made up of members of government as well as industry. And this was a very important collaboration. Now, our terms of reference was simply provided operational context, how we did things. Now, the more serious documents were the research prospectus, and within that prospectus, we had research themes. And under each of the themes that are listed here, 1 through 4, each of them had a series of questions, and we called them critical questions, but in reality, they were priorities that fell into mountain pine beetle biology, the hydrological impacts of the mountain pine beetle, landscape and stand dynamics, and the social and economic implications of a changing landscape. And through these 4 research themes, 38 research projects were supported from 2007 to present. And I also have to suggest and emphasize that the funding for these projects was received entirely from the government of Alberta. So I just want to wrap up by just issuing a few parting thoughts, and just to emphasize the importance of the research that is being done on mountain pine beetle. Nearly 2.4 million hectares, 44% of the leading pine stands are variably infected by mountain pine beetle. The economic value of pine resources is also estimated at about $11 billion, which translates into economies that sustain forest communities, and it's so important that we manage the mountain pine beetle outbreak and in order to sustain the forest industry, to maintain economies, but also to address the diminished supply of ecological services. We don't know exactly what the cost is of the decline in ecological services, but we have to presume that it's rather significant. And so there's an expanding role for science-based solutions. And this is in contrast with a lot of things that we hear about COVID virus and being able to control it, but really our actions in Alberta with respect to mountain pine beetle have to be science-based. So this webinar series is going to provide snippets of the research conducted to manage and control the spread of the beetle. And I hope you enjoy all of the webinars. But I also would like to take just a brief moment here to say that Alberta owes a debt of gratitude to all the scientists and all the practitioners who have dedicated time and intellect to the management of the mountain pine beetle. Now, what I would like to do is to introduce Antonia Muso. And Antonio is a PhD student working in Maya Evidence Lab at the University of Alberta. And it's interesting, Antonia, you could read her bio in the information that is provided for this webinar. But Antonio started her work at Simon Fraser University studying a rodent chemical ecology. And this is where she did her master's. And as a result of, I guess, taking a course on forest pest management, she felt that really her real true venture in life, or well, she uses love of life. And I understand Antonio just got married, so I don't want to use that sort of glibly, but is entomology. And so Antonia moved to Alberta to study with Maya Evident, and she is just in the midst of completing her PhD thesis. She has a little bit more data analysis to do, but we wish her all the best in bringing that to a conclusion. So what I'd like to do now is turn it over to Antonia, and I'd be happy at the very end to answer any questions that you may have. But really, as I indicated earlier, the real feature is Antonio. Antonia, take it away.
Thanks so much, Keith. Entomology is my truest love. I'm pretty sure my partner would be okay with me saying that. I'm just looking at, oh, my microphone is working. Perfect. Okay. So thank you everybody for coming. Hopefully everybody's having a good day. So today I'm going to talk to you about my first chapter of my thesis that was partially funded by the TRIA Network as well as FRI. My title is Pine Wars, A New Host. And if you are a Star Wars fan, you get that. What about mountain pine beetle? So climate change has facilitated the range expansion of not just mountain pine beetle, but many forest insects all over the world. So mountain pine beetle is just an example of this. And of course, in the province of Alberta, we know that mountain pine beetle has continued to expand after the massive inflights, as Keith explained in 2006, and spread across the province. It's definitely has made its way into the boreal forest. So my sort of aim when I joined U Alberta was to ask some questions about how mountain pine beetle might be, how its biology is changing in the expanded range and its risk of spread into the boreal forest. So you're all probably, can you see my cursor if I put it here? I don't know if anybody can say yes. So everybody's familiar with the mountain pine beetle life cycle. The part of the life cycle that I am going to focus mostly on, and what my thesis mostly focuses on, is the dispersal portion of the life cycle, as well as the colonization of new hosts. So this is the part of the life cycle that I'm focusing on, and today I'm going to be talking mostly about mass attack, which is of course the characteristic behavior of the incipient epidemic and epidemic population states. So during this time, mountain pine beetle is at high density on the landscape, and they're able to attack vigorously defended trees. So during host colonization, mountain pine beetle need to contend with the tree's constitutive and induced defenses, and these are both physical and chemical defenses. So like the beetles in this video, they're met with sticky resin that contains toxic chemicals and that the tree is trying to prevent them from colonizing. So on their own, a beetle can't colonize and reproduce within a tree. They need to mass attack. And to do this, they use pheromone aggregation pheromones, and that enables them to overwhelm these healthy, vigorously defended trees, which is great because these trees are very rich in resources and are good for brood development. So when looking at mass attack, there are two critical thresholds that are important for mountain pine beetle's success. So the mass attack threshold is the minimum density of beetles that enable successful mass attack. And we know this from studies done in the historic range that this is approximately 40 attacking beetles per square meter. And then past the mass attack threshold, there is an optimal attack density. So brood production continues to increase until a peak optimal density, which is about 60 attacking beetles per square meter. And then once they start attacking more than 60 per square meter, brood success begins to decline because of intraspecific competition under the bark. So you have lots of larvae living under the bark and they run into each other and will run out of resources. So our question was, how might mass attack dynamics be different in the expanded range from the historic range? We know from previous research that pines in the expanded range, both lodgepole and jack pine, have fewer defenses. And on top of that, pines in Alberta are more likely to experience drought stress, which potentially makes them more susceptible to mass attack. So if you know jack pine, it grows in really sandy soil, which is very well drained. So jack pine has pretty low water availability. So this potentially would make it more susceptible. So the black line on the graph is the mass attack dynamics based on data collected in the historic range. But we predicted that the mass attack dynamics in the expanded range might look something more like the red line, where there's a lower mass attack threshold and a lower optimal attack density. So we set out to test these hypotheses. So the reason why we wanted to do this is there's some pretty big implications for there being a lower mass attack threshold. It could possibly impact endemic epidemic dynamics. So if it takes fewer beetles to mass attack a tree, you might see more frequent outbreaks. And attack density is also currently used in management to determine which trees are controlled. So currently the threshold is 40 attacks on the tree bole gets marked for control. So when testing this, we did it both in lodgepole pine. So we did it over 2 years in lodgepole pine in 2015 and 2016, south of Grand Prairie. And then we did it in jack pine in 2018. So first, I'm going to talk about the lodgepole pine research and then switch to jack pine because the methods were a little bit different. So in lodgepole pine, in each year, we baited 15 trees at one site. So we tried to get 5 replicates of 3 different treatments. So we had a low attack density treatment, which was 20 attacks per square meter, a medium attack density treatment, which was 60, and a high attack density treatment, which was 100. So what we did was we baited those trees with aggregation pheromone, and then we monitored them every 1 to 3 days during the flight period. And once mass attack had begun, we watched those trees during the day to make sure that we could stop the attack at the desired cutoff point for the treatment. And to stop attack, we wrapped the lower 3 meters of the bowl in wire mesh to prevent beetles from coming in. So when we actually harvested these trees, we harvested the center meter of the bowl. So we harvested from 1 meter off the ground to 2 meters in height. So the screening we removed in the fall, and we let the trees stand over winter because we wanted them to be as natural as possible. So in the following spring, typically around May, we harvested 2 50-centimeter bolts, like I said, about 1 meter off the ground, and we brought those back to the lab. So we placed the bolts in emergence bins and collected the emerging beetles, and then we measured those beetles and counted how many came out. And then once emergence had finished, we autoclaved the bolts and traced the galleries onto clear plastic. So the bark of lodgepole pine doesn't come off in a big, nice chunk. So we had to peel the bark and then put clear plastic over top and then trace the galleries. And then we quantified those galleries using ImageJ. So when looking at the results from this study, lodgepole pine, this is the number of larvae per female that were produced. So when we harvested the 2015 experiment, we were super shocked because the low attack density treatments actually had the highest number of larvae. And we were like, wow, this is like crazy, because that seems like such a low attack threshold, being around 20 attacks per square meter. So that's why we decided to repeat the experiment in 2016, but our results didn't hold up. So we didn't see the same success at 20 attacks per square meter, and we actually saw better success between 60 and 80. So this is the larval survivorship. Past that, we also looked at the number of pupae and adults that emerged. So again, it makes sense. Where there were lots of larvae, there were lots of adults. But then when you look at the curve for 2016, there were the most pupae produced at around 80 to 100, which is the complete opposite of what we got the year before. So it's all very confusing. So from the lodgepole pine work, the relationship was different in the years tested. The 2015 data supported our hypothesis that there's a lower mass attack threshold. However, 2016 had a trend comparable, or if anything, potentially even higher than the historic range. But the number of brood per attack was very low. So if you look at the number of brood per attack at these low densities in 2015, there's around 9 offspring per female produced, whereas our max offspring per female produced was only 2 in 2016, which is quite low. So there are a few reasons why this could be. So it's possible that the beetle pressure was not as high in 2016. So fewer beetles were in the area, and the mass attack period was much longer. So when I was going out and recording how many beetles were on the tree at each day, it was really slow. So some of the trees took up to 2 weeks to reach their desired density. This was definitely contrasted to 2015, where once the beetles started attacking, they would reach their desired attack density, say 60 attacks per square meter, within 24 hours. So there was definitely an extended attack period. I'm not sure whether this was because there were just fewer beetles in the area. The other thing that was different between the years was 2016, during the flight period, had 2 times the number of rainy days, which interrupted the flight period. So mountain pine beetle really don't like flying in the rain. So these extra rainy days could have also resulted in the attack period being extended. So there are a couple reasons why this could be. I repeated the experiment again in 2017, but woodpeckers ate all the beetles over winter. So this is, I guess, just the way science goes. Sometimes you win some, you lose some, I guess. So I think potentially our conclusions from this could be that attack success is definitely dependent on weather as well as beetle pressure. So in years where there's lots of beetles and it's quite dry, you might see a lower attack threshold than in years where it's wet and there aren't as many beetles. So we also wanted to test this in jack pine, and in those same years that we did the lodgepole pine experiment, we also tried to get mass attack in jack pine. So we did that in 2015 and 2016 by baiting trees, and not enough beetles seem to be in those areas to get mass attack in more than a few trees. So we needed a lot of replication. So only 2 trees getting mass attacked was not good for us. So we tried seeding jack pine trees with infested bolts, where we added bolts underneath the trees and baited the trees in hopes that the beetles would come out and attack the tree, but that didn't work either. The bolts produced unreliable numbers of beetles, and they just didn't go into the jack pine. So in 2018, I was kind of done with trying the bolts and trying to get natural beetles. So I decided to mass release actual beetles underneath jack pine trees to try to artificially increase the beetle pressure. So this was done in 2018 north of Lac La Biche. So definitely jack pine forests. So the way that we did this was to get these beetles to release under the trees was we harvested approximately 80 infested bolts from the Hinton area, which in 2018, there were lots of infested trees. And we cut down about 30 trees that summer and loaded up 3 trucks worth of bolts. And we brought those back to the lab, and I filled our teaching lab at the university with all of these bolts and mass reared about 50,000 mountain pine beetle that summer. So once the beetles emerged, they were counted and put into buckets with jack pine wood chips. So we were hoping not only releasing the beetles with a baited tree, we also wanted them to have an early adult experience of jack pine, potentially giving them kind of a taste for it. So we ended up manipulating 18, well, actually, that's not true. In total, there were 26 trees. Only 18 of them were harvested. The other 6 were left to overwinter. So across 3 sites. So these were north of Laklavish again. So each manipulated tree was fitted with this wire mesh tent. So it was secured just above 2 meters on the bole so that the lower 2 meters of the tree were exposed to the released beetles. The trees were also baited with aggregation pheromone and host volatiles to further convince the beetles to attack the tree. And then what we did was we put one of those buckets with 1,000 mountain pine beetle in it under the tent and then sealed it up. So we buried the edges of the tent in the sand and to prevent the beetles from escaping. So in order to try to get a range of attack densities, we put the buckets under the tree and allowed the beetles to kind of naturally diffuse out of the bucket. And for trees that we only wanted a low attack density in, we removed that bucket after 24 hours and then put the remaining beetles that hadn't come out of the bucket into the tree skirts of the trees that we had designated high attack density. So those were assigned at random. So if the bucket was removed after 24 hours, it was assigned low. If the bucket was just left under the tree, it was assigned medium. And if it had an addition of a bucket after 24 hours, it was assigned high. So we tried to release these during the sort of regular flight period for mountain pine beetle in Alberta. So site 1 was on the 15th of July, site 2 the 1st of August, and site 3 was on the 10th of August. So in October 2018, before overwintering, we wanted to harvest a subset. So we ended up harvesting 2/3 of the trees and took the 2 50-centimeter bolts that we usually do from each tree. So those offspring were allowed to complete their development in the lab and then were collected in our emergence bins. And once they had all finished emerging, I autoclaved those bolts in our giant autoclave in the basement of the biocide building, and then peeled the bark off. So because jack pine has much thicker bark, we're actually able to peel the bark off and get the gallery characteristics on the inside of the bark. But if the bark didn't come off as a nice, beautiful piece, we then did the same thing like we did for lodgepole pine, where we traced the gallery characteristics onto clear plastic and then quantified those again. So this is what the center picture is the inner bark of a jack pine tree. The image on the very left is a typical what the inside of the bark looks like in lodgepole pine. So you can see the vertical parental galleries in red, and they make oval pupal chambers coming out from those parental galleries. So when I peeled my first jack pine log, it was this one right here in the center. I had no idea what I was looking at because I didn't see these characteristic oval pupal chambers. I saw a few larval galleries, but then where I expected to see the oval pupal chambers, excuse me, was this huge area of consumed phloem where all the pupal chambers seem to have fused into this big area of consumed tissue. So the upper image here is what, on the very right, is what a trace onto plastic looks like in lodgepole pine, again with those characteristic oval pupal chambers. But then this on the bottom is what it looked like in jack pine, where you have all of the area that's consumed filled in with marker. So possibly why this is happening is because jack pine has much thinner phloem on average than lodgepole pine. So in my lodgepole pine experiments, the average phloem thickness was 2.3 millimeters. However, in my jack pine experiment, the average phloem thickness was only 1.5 millimeters. And when I selected my trees to manipulate in jack pine, I only selected trees that had a greater average phloem thickness than 1 millimeter. So lots of jack pine trees have thinner than 1 millimeter phloem. So what could be happening is mountain pine beetle needs to mine a larger area to get the same amount of nutrition as they do from lodgepole pine, since the phloem layer is thinner. Rather than mining the characteristic shapes, they have to mine wider larval galleries and wider pupal areas to get the same amount of nutrition. So when we quantified the number of brood at each attack density, so the first thing I was really excited about was that my method for trying to get a variable attack densities actually worked really well. So we had attack densities anywhere from quite low, so only like 9 attacks per square meter, all the way up to really heavily attacked, which were around 180 attacks per square meter. But when we draw a curve around the data, we see that trees as low as 20 attacks per square meter have successful brood development, and the peak seems to be somewhere around 40 attacks per square meter. And so the gray lines here are just a reminder of the at 40 attacks per square meter is the mass attack threshold for lodgepole pine, and then at 60 attacks per square meter is the optimal. So you would expect to see the peak at 60 in lodgepole pine. So it looks like the peak is indeed lower in jack pine. However, we did our best to count the number of distinct pupal areas with the tree, with the fused pupal chambers. I think that the height of this curve is underestimated because there could be more beetles coming out of these fused chambers than we were able to count. So one of the things that I'm doing to try to amend this is calculating the percent area of the underbark environment that is consumed, and then cross-referencing that with the number of actual beetles that emerged. So I was in the process of counting those beetles in my freezer, and then COVID happened. So I am hopefully going to be able to finish doing that before the end of this year. So another thing that we looked at was offspring condition. So we were interested to see if attack density, since at high attack densities, there's more competition, potentially there would be less good quality of offspring coming from high attack densities. So to do this, we calculated an offspring condition residual index. So basically what we did is for each beetle that emerged, we took a subset of 50 beetles from each, 50 males and 50 females from each attack density or each bolt. And we got their body size by doing surface area. So we got the pronodal width as well as the body length. And then we also did fat extractions where we used a sock slit apparatus to extract all the fat from the beetle. And we weighed them before the extraction and after the extraction. So the difference in weight would be the amount of fat that they had. So to create this condition index, we took the body size of each beetle and regressed it against the fat content and then fit a linear model to that. So, and then we got the residual value for each of these beetles. So a beetle that has a positive residual has a better body condition than expected from the model, and a beetle with a negative residual has a less good quality than we expect based on the model. So when we analyzed this, we saw there was, interestingly, a significant interaction between density, attack density, and sex. So females, you see no difference in body quality as attack density increases. And where the interaction comes from is males actually increase in body condition with increasing attack density, which is the opposite of what I expected. I expected there to be a decrease in body condition. So something could be going on here where at high attack densities, perhaps there's lots of fungus and the males are getting lots of nutrition that way, and they are increasing their body condition at high attack densities. And of course, the difference between the lines is because females always have a better body condition than males. Another significant part of the model was phloem thickness. So as phloem thickness increases, the quality of the beetles also increases. And this makes sense because the more phloem, the more resources, so the better body condition they have. Interestingly, another thing was there was a marginal effect of an interaction between density and phloem thickness. And what this looked like was trees with thicker phloem also had higher attack densities. So could be a few reasons for that. It could be that the thicker phloem trees result in beetles producing more aggregation pheromones. So there are more beetles attacking those trees. It could just be that trees with thicker phloem are just able to support more beetles, so more beetles attack that tree. So I would have to do some more experiments to figure out exactly what's going on there, but nonetheless, I think it's interesting that thicker phloem trees have better quality beetles. So conclusions from the jack pine work is that our data support the hypothesis that the mass attack threshold is lower in jack pine. So it takes fewer beetles to kill a jack pine tree. The optimal attack density is also lower, but I think it's less clear as the optimal attack densities produce more beetles, but also have the large fused pupal chambers. So I'm not super confident in my ability to quantify the number of beetles. So again, that height of that curve, I think, is probably higher than what I have estimated at this point. And thin phloem means that there's more area under the bark consumed in jack pine. So the reason why I think that's interesting is at high attack densities, maybe there's even more of a limit on the number of beetles that you can have in a tree. If an optimal number is consuming all of the phloem, then past that point, there wouldn't be enough phloem, and at high attack densities, even fewer beetles are being produced. So I'm still kind of playing with some of this data and working on it. Again, I'm working on estimating how many offspring emerge with different percentage of underbark area consumed. So what that looks like is me taking these photos of the inner bark peels or of the plastic traces and outlining everywhere that's consumed, and then calculating the area and dividing that by the total area of the bark trace or the log surface area. So that's taken a little while, but I'm just about finished. The other data that we took from this experiment was tree chemical response to variable attack densities. So prior to mass attack, so either prior to baiting the tree or prior to releasing the beetles, I took phloem samples from each of those trees and put them on dry ice. And then I took another phloem sample after mass attack to see whether which volatile chemicals were being upregulated. And then I also took another phloem sample after the overwintering period to see kind of the legacy of mass attack on the monoterpene profile. So those samples were ready to send in March, but then the analytical lab that was going to do the analysis shut down. So I sent them in June, but they are hopefully going to be finished analyzing them because they had a big backlog due to the shutdown They should be done analyzing them by the end of the year. And so I should have that data really soon, which I think will be really interesting. So again, just a reminder of the implications. So lower mass attack thresholds could impact endemic epidemic population state dynamics in the expanded range. So if it takes a lower density, this curve could also be moved this way as well, where a lower density of beetles results in the incipient epidemic population state, and then the swing into the epidemic population state. And again, attack density is used in management to determine which trees are controlled. So the current control threshold of 40 attacks on the bole is already very conservative. So I don't know if it needs to be changed based on this data, but I think giving the data to the province will allow them to decide what is the right decision and what is the right threshold for looking at this kind of stuff in jack pine. So all of my photos from my acknowledgement page have disappeared. I had a glitch when saving my presentation about half an hour before starting. I accidentally unplugged my external hard drive, which glitched my presentation. So all of my photos are gone. So I'm just going to have to explain to you. So I want to thank the Edmonton lab members and our 2 technicians that worked with me during the time that I was doing these experiments. I had 3 amazing undergrad assistants that helped me with this field work, and it would have been impossible without them helping me put cages around trees and rearing beetles. They were amazing. And I want to especially thank Victor Shigelski, who cut down those 30 trees that we used to rear those beetles for the jack pine experiment. And there was an Alberta Agriculture and Forestry logo here. It is now gone, but I want to thank Caroline for helping me get all the permissions to do all of this. Devin in Grand Prairie and Fraser in Lac La Biche for helping me pick sites and get all the permissions that I needed to. And the pictures that are supposed to be here are TRIA Network, University of Alberta, NSERC, and FRI. So all the jack pine work was funded through an FRI project. So thanks for listening to my talk. I hopefully left lots of time for discussion and questions. So thank you.